Hello, and welcome to my presentation on the current SARS-CoV-2 testing forms. First, what is coronavirus? As the name suggests, this disease is part of the virus family. Viruses infect other organisms by inserting their viral genetic information into a host cell. Once the cell is infected, it will start producing viral proteins and viral genetic information, increasing the virus population. In theory, once a cell is infected, it cannot become uninfected. Instead, the immune system produces antibodies to bind to the virus molecules and prevent them from infecting more cells. We are able to see the structure of a generic virus molecule depicted on the left. There will be some type of genetic material enclosed in a capsid, sometimes RNA and sometimes DNA. We will also have an envelope and proteins, which are represented by the spiky knobs. If we look at the coronavirus molecule on the right, we can see a grayish envelope covered in red spiky proteins, and although it's not depicted, there is genetic information on the inside. More specifically, the genetic information is RNA. For reference, human cells have DNA, which is typically double-stranded, whereas the coronavirus molecule has RNA, which is typically single-stranded. This is notable for the upcoming information on tests. There are currently 3 types of tests used by medical professionals for the coronavirus. Here we can see the names of each test, as well as each testing medium. The first 2, molecular and antigen, are diagnostic tests. These tests are able to identify an active case of COVID-19. The third test, the antibody test, is a serology test. A serology test is used to identify antibodies to COVID-19, which means these types of tests are not capable of diagnosing someone with an active case, but might be able to identify if a person has been infected in the past. The first test we will look at is the molecular test. This test often receives the title of gold standard since it's the most effective at identifying an active coronavirus infection. The sample type is often a nasal or throat swab, but recently saliva has been used. In fact, saliva is currently the testing medium for the UW diagnostic tests. The method used to detect the virus is named reverse transcriptase polymerase chain reaction, or RT-PCR for short. More on this in the next slide. The main benefits of using this test include rare false positives and a high effectiveness for symptomatic patients. Some downsides of the molecular test are the price point and the wait time before receiving results. Both of these pitfalls are due to the special equipment required to run the RT-PCR tests. Usually, it's expensive and located away from the testing site. As mentioned, the molecular test uses RT-PCR. PCR, without the RT, is a technique used in many diagnostic tests to amplify DNA by making millions of copies. The steps of PCR are represented by the little clips on the right. Now, you might remember that coronavirus does not have DNA, it has RNA. To account for this, an enzyme named reverse transcriptase is used in the process of converting RNA to DNA. Now that we have DNA, we can proceed with the normal PCR procedure. The first step is to heat the DNA solution to separate the 2 strands. Next, primers are added to aid in the synthesis of DNA. In the third photo, an enzyme is able to elongate the single-stranded DNA back to double-stranded DNA. We now have twice as many strands of DNA as before. As this process is repeated over and over, marker labels attach to the viral or target genetic material. These labels will give off a fluorescence that the computer can read and identify as an active coronavirus case. A unique feature of PCR tests is their ability to provide insight on the level of infection. If the fluorescence is observed in fewer cycles of PCR, we can note the infection is more serious. The other type of diagnostic test is the antigen test. For reference, an antigen is any type of substance that causes the production of antibodies. This test uses a nasal or throat swab to detect an infection. Currently, the most popular method involves the use of a lateral flow assay. Assay and immunoassay are simply defined as a procedure for identifying a specific substance in a solution. Moreover, my research mentor, Patrick Johnson, and his team of students are also engineering a SARS immunoassay. The major benefit of antigen tests are their rapid result times, usually in the range of 15 minutes, as well as their price point and their portability. The major drawback of these tests is that a degree of sensitivity is lost when compared to the molecular test. The first method of antigen testing is the lateral flow assay, which uses antibodies to detect antigens. It's important to note that antigen tests use antibodies, but they are not an antibody test. The antibodies used in antigen tests are lab-made to aid in the diagnosis of a given antigen. Here's some helpful vocabulary before moving on. An analyte is the target substance, specifically in our lateral flow assay, and the antigen is a specific type of analyte that is able to bind to antibodies. In a lateral flow assay, the sample is prepared and placed on a test strip where it moves across the strip. If the target antigen is present, it will bind to a fluorescently labeled antibody. The sample continues to move down the strip. If the antigen-antibody combination is present, it will bind to another antibody on the test line, resulting in a color change. The control line will also change color if it is able to bind to fluorescently labeled antigens common in human samples. In theory, if an adequate sample is taken, this line should always change color. The three scenarios are positive, where we can see a control and test line, negative, where we can only see a control line, or invalid, where that control line is not visible, meaning an adequate sample was not taken. The most common lateral flow assay is the pregnancy test. In this case, the target analyte is the HCG hormone that is secreted during pregnancy. This hormone has antigenic properties, which allows it to bind to antibodies for the purpose of this assay. The SARS immunoassay will use a similar model to the lateral flow assay with the sandwich complex. SARS stands for Surface Enhanced Raman Spectroscopy, which is a technology used to identify given molecules by a qualitative graph called a spectrum. Every molecule will produce a unique spectrum, which can be thought of as the molecule's fingerprint. This SARS immunoassay will actually be an indirect test, meaning we are not looking for the spectrum of the coronavirus antigen. Instead, we will use a specific Raman dye to coat the gold particles and look for its spectrum. In this test, an antigen will bind to two unique antibodies, one on a gold nanoparticle and one on a magnetic nanoparticle, forming a sandwich complex. Finally, a magnet will be used to pull all of the magnetic nanoparticles and any sandwich complexes together, if the virus is present. A laser will shine through the collection of particles at the magnet, and the Raman device should display a spectrum matching the Raman dye that is coating the gold particles. If the virus is not present, all of the magnetic nanoparticles will still gather at the magnet, and a laser will be pointed through them. However, since there are no gold nanoparticles, the Raman dye will not be present to produce the corresponding spectrum. Here are an example of the spectrums of a given Raman dye. The final type of test used in coronavirus studies is the antibody test. The most common sample for this test is a blood sample. A modified lateral flow assay can be used to detect coronavirus antibodies. Also, an ELISA test can be used for the antibody detection, but I will not be going over the specifics of this test. The major and obvious benefit of antibody tests is that they can be used to understand the spread of the virus by identifying a previous case of coronavirus, even if the person was asymptomatic. Also, if a person is found to have coronavirus antibodies, they could donate blood for vaccine development purposes or to sick patients. One interesting downside of the antibody test is that there could be a false positive if a patient has antibodies to another strain of coronavirus. In other words, antibody tests are not yet specific to the 2019 strain alone. Finally, due to cases of reinfection, it is known that even if a person has coronavirus antibodies, that does not guarantee immunity. As companies around the world improve testing, it is clear that there is not a perfect test or a perfect treatment for this virus. With this in mind, please do your part in decreasing the spread of COVID-19. Wash your hands, wear a mask, and stay home if you feel ill. Thank you, and stay safe. The Wyoming Research Group also works closely with a team from the National University of Ireland, Galway. This project is funded by the Health Research Board in Ireland.